Physiology's most beautiful equation, as I'm calling it, is basically a relationship between ventilation and perfusion in a gas exchange organ. We'll develop that equation more explicitly in a little bit, but before we do, it will help if we understand something about how a specific gas exchange organ works. Our example is going to be the fish gill. Let's start with the basic lay of the land. The fish gill sits at the periphery of the fish's pharyngeal cavity and is ventilated by drawing water into the mouth, forcing it backwards over the gills and out into a peripheral space called the opercular cavity. The water is then expelled through the flap-like opening in the side of the fish behind the head called the operculum. Looked at from the front, the gills are arrayed on either side of the pharyngeal cavity. Water flowing into the mouth is distributed to each side of the pharyngeal cavity and over the gills. Each gill is built upon a cartilaginous bar that runs dorsoventrally, called the gill arch. These are arrayed in pairs on either side of the pharyngeal cavity, and in a typical teleost fish, there are 5 or 6 of these pairs of gill arches. The gill arches serve largely as a structural support for the actual gas exchange surfaces, which are located on a series of paired leaf-like evaginations called gill filaments. The number of these gill filaments varies depending upon the size and lifestyle of the fish. Active, fast-swimming fish typically have filament numbers in the thousands, whereas slow-swimming, sluggish fish typically have filament numbers in the hundreds. Larger fish obviously have more gill filaments than do smaller fish, and so on. The rules are pretty intuitive. Gas exchange across the gills does not take place on the gill filaments themselves, however, but from a further set of evaginations emerging from the gill filaments, thin membranes known as gill lamellae. These are arranged along the length of the gill filament on both the filament's upper and lower sides. Here's an electron micrograph of what they look like. In this image, the preparation has caused the lamellae to collapse against one another, but in real life they are separated from one another along their length, held apart by the water that courses between them. The lamellae are very densely packed along the filament, separated from one another by distances ranging from about 25 micrometers between filaments up to a maximum spacing of about 100 micrometers. These have the effect of increasing the surface area for gas exchange tremendously. Again, this enhancement depends upon the body size and lifestyle of the fish. Sluggish fish have total gas exchange areas of a few hundred square centimeters. A large actively swimming fish, like tuna, on the other hand, has a total gas exchange surface area of nearly 24 square meters. Generally, gill surface area scales with body mass to an exponent ranging from between 0.7 and 0.8, pretty close to what we would expect for the scaling of metabolic rate. Let's look now more closely at the structure of the gill arch and gill filament, and focus in particular on the pattern of blood perfusion through this structure. Here's a cross-section through a single gill arch. Let's quickly orient ourselves. The mouth is located to the top of this diagram and the esophagus at the bottom, and the pharyngeal cavity is sandwiched between them. We've oriented the pharyngeal cavity on the left and the opercular cavity on the right. Each gill arch has a pair of rigid ridges along them known as gill rakers. These act to deflect large particles from moving through the gills, which could potentially clog or damage them. The paired gill filaments project from the gill arch into the opercular cavity. The gill arch is perfused by 2 arterial vessels, and in both, blood runs from ventral to dorsal. Blood fed from the heart into the fish's ventral aorta is fed into an afferent gill artery that conveys blood up the gill arch. This is deoxygenated blood. This blood is then fed into the gill filaments through arterioles that run along the back edge of the gill filament. Blood is drained from the gill filament along the filament's anterior edge, eventually feeding into the so-called efferent gill artery, which feeds into the fish's dorsal aorta. This blood is oxygenated and is directed to the tissues of the body. Gas exchange takes place between the water flowing between the lamellae and capillaries arrayed within the gill lamellae. The important point to note here is the general direction of blood flow from the filament's posterior rim to its anterior rim. This becomes important when we consider the direction of flow of water through the gills, which runs from the mouth into the pharyngeal cavity and then out through the opercular cavity. In short, the direction of water flow between the lamellae is opposite to the direction of blood flow through the lamellae. This is known as a countercurrent exchange system. This structure and pattern of blood flow and water flow, of perfusion and ventilation, to state it properly, underlies one of the most efficient gas exchangers that exists in the animal kingdom. Let's turn now to exploring how this gas exchanger works.